I suggest to start, even though I'm sure there are still colleagues that will join or interested participants. I have just some introductory slides for ICPerMed for those that are maybe new to our webinar series. So before we are coming to the main presentations today, I would like to take the occasion also to introduce a little bit our webinar series in general and ICPerMed. Yeah, so welcome everyone to our webinar series from the International Consortium for Personalized Medicine. Our aim is through different webinars to introduce or to share different international perspectives on personalized medicine. So we hope via these webinars to go over the entire globe and to see how different countries and different organizations are pushing forward the field of personalized medicine. So my name is Monica Frenzel. I'm working at the French National Research Agency, and I am the Internationalisation Working Group lead within ICPerMed. So very few words on ICPerMed itself. ICPerMed, as I said, is the International Consortium for Personalized Medicine, and its members are mainly regional national ministries of health or research, of funding organizations of the respective countries, as well as policy organizations, and the European Commission is also part as observer in ICPerMed. Our overall aim is to coordinate and promote research along the entire value chain and together to develop and promote personalized medicine. We're having two working groups within this consortium. One is the working group on internationalization. In this frame, this webinar is organized, and also the policy and strategy working group. What we are doing is specifically this working group. We support the international outreach, specifically on the policy level, but also we would really like to better understand what is going on in the field of personalized medicine in the different countries. Our main aim is to collect and to share knowledge, for example, through this webinar, and also to bring different initiatives to work together over the entire globe. And sometimes this webinar could be a good occasion to learn what others are doing and to get new connections. So there are just some examples. So we are doing interviews and webinars, and also through the website, we are promoting all this, including also achievements and activities of individual countries. And also we are promoting good examples of personalized medicine, especially when it comes to implementation. So research that finally made all the path towards implementation in a system of health. And I feel that today's talks that are really also outlining the structuring efforts that could take place in a country in a centralized way. This is really interesting and part of this idea of sharing good practices in the field of personalized medicine or activities that will support personalized medicine. And now it's really my immense pleasure to introduce the two speakers we have for today's webinar. And the overall topic of the webinar today is genomics, infrastructure, and AI-enabled omics. And we have two speakers today, Graziano Pesole, who is professor of molecular biology at the University of Bari Aldo Moro, associated research director at CNR IBIOM. You have always to correct me afterwards if this was all correct, the way I pronounce this. And also very important here for the structuring effort, head of the Italian node of Elixir. And Dr. Pesole will specifically introduce as topic in his talk the European 1+ Million Genome Initiative and the Genomic Data Infrastructure. And then our second speaker today is Dr. Bruno Fosso, Associate Professor in Molecular Biology at the University of Bari Aldo Moro and co-lead of the Elixir IT Italian tools platform, if I translated correctly. And Dr. Fosso will specifically concentrate in his contribution today on the AI-driven multi-omics for microbiome investigation. Yeah, thanks a lot to both of you for joining today. If you would like to learn more about the background of our both speakers, don't hesitate to have a look on our website, where you can find also a more detailed bio of both speakers. And therewith, well, already thank you for joining today's webinar. I will stop my presentation, and I would like to hand over to Professor Pesole for starting today's webinar and, well, your part of the presentation. Thanks for joining. It's a real pleasure to have you with us.
Thank you very much, Monica, and thanks for the nice presentation. As anticipated, I will try to provide you a summary overview of the 1 Million Genome Initiative in Europe and all the effort to build the genomic data infrastructure, with a special focus on what we are trying to realize in Italy. So I would like just to start, okay. So, of course, precision medicine, now we understand very well, is critical for national health system sustainability. We have several challenges. The first one is that scientific research is continuously developing new drugs and therapies, and these are expensive. And of course, we have to guarantee the best cure to everyone. And this, of course, conflicts with the sustainability of a system. The second one that we know very well, that is a good thing, that is not so good for so the balance of a national health system, is that life expectancy continues to increase, and this leads to a higher age of the population, with a greater incidence of age-related disease. And finally, as you know, natality is lowering very deeply in these last decades, and so the ratio of retirement workers increase as life expectancy rise, and this threatens the efficiency of the national health system. The plot that you can see is a plot of life expectancy across the years. So it's trivial to say that precision medicine, real precision medicine, represent the only solution for maintaining the sustainability of the national health system, while harnessing scientific advance to improve population health and promote healthy aging. So this what requires? This requires a very critical changes in organizational, regulatory, and technological aspects that involve generation and sharing of big data that, of course, include genetic profiles and associated clinical and personal metadata. So all of this descend from what we call the genomic revolution that has been, the result of the advent of high-throughput sequences since some decades since now. By the way, we initiated to the sequencing era about 50 years ago. That you can see in the bottom a very aged paper on the mitochondrial genome. And of course, a landscape breakthrough was the completion of the first draft of the human genome that, as you know, took 13 years for over $10 billion. And now we have the capacity to produce a human genome in few hours and a very cheap cost. So you can see in this slide the two covers. The first one is the Nature paper in 2001. That was the first draft of the human genome provided by the Human Genome Consortium. And then the last draft, that is the so-called T2T genomes, that was published in 22. And as we know, the essence of each individual is enclosed in its genetic material that involves about 3 gigabase pairs on 23 pairs of chromosome, and another teeny genome that is the mitochondrial genome, which involve about 6.5 kilobase. And all of this is codes for about 20,000 protein coding genes and about 43,000 non-coding RNA genes. So in general, the genome of 2 individuals is very homogeneous. Human population is genetically very homogeneous, sharing 99.9% of the genome sequence, but still there are over 4 million differences between 2 different individuals. The real big breakthrough in this adventure has been the development of next generation sequencing, starting from the traditional Sanger sequencing. In order to drive this transition, we have seen 2 real evolution. One is the fact that we don't need the cloning phase to sequence the DNA fragment. And so the library preparation is just made by linking adapters to the unknown fragments. And the second important evolution has been the possibility to obtain high throughputs by high-level parallelization of sequence reading. That is the fact that we don't need anymore the use of electrophoresis separation in order to detect the sequence. So, of course, there has been a terrific evolution in the sequencing platform. As you can see in these slides, in the x-axis, you can see the red length, and in the y-axis, you see the throughput of the production with all the platform that has been generated and commercialized over time. And I report here the state-of-the-art platform, like the NovaSeq X Plus platform that is able to produce about 16 tera base pair per run with over 100 billion reads. And in the field of long reads, we have essentially 2 technologies. That is the Oxford nanopore technology that is able to produce over 6 tera base pair, which are very long, or over 30 kilobase long, that I will discuss this later, or the PacBio platform that again is able to generate also long read. So in general, we experience a continuous increase of the volume of data that we are able to produce, which a never decreasing cost per base pair that is now at about 2.5 euro per gigabase. So very briefly, the 2 state-of-the-art platform are the Illumina sequencing platform that use the typical termination-based strategy, which blocking groups which are able to emit, we are able to read by fluorescent assay 1 base at a time. And this also guarantees this high throughput that I showed you before. And more importantly, in my opinion, this is a really breakthrough in sequencing platform in the Oxford nanopore technology, which offer presently the unique possibility to sequence both RNA and DNA directly, and is also able to detect in real time nucleotide modification. And as you know, these are very informative for the biological information, mostly at level of RNA than at DNA. That, as you know, we have in DNA the methylation that, of course, impact on the epigenetic profile. And this is achieved by crossing the DNA filament or RNA filament through a large set of nanopore, where we are able to detect current variation. these current variations are interpreted by AI software, very sophisticated software, in order to decode this electric signal as a sequence. And is also able to detect not only native sequencing, but also modified nucleotides. The limit of this technology is that it's not able to detect one nucleotide at a time, but detect a packet of k-mers, and this, of course, involves an higher error rate that can be minimized by increasing the coverage. So this slide, in my opinion, is really very interesting. It shows that what we are experiencing now is that we increase to both an increase of storing technology, so the hard disk costs less and store more, but also the sequencing platform are able to sequence more at a lower cost, but the growth rate of data production is twice that of storage capacity. That, of course, face us to a real problem in the future. So we need, very sophisticated technology to be able to continue to store the data. So we have to invent something more in the future in order to parallel these two needs across time. So now we go to how to do genetic profiling. As I told you before, we can follow two different strategies. The common strategy is to use the so-called short reads. So the state-of-the-art technology for this is Illumina, as I mentioned you before, or long reads. But in any case, what I can say is that at the moment, the sequencing cost is lowering so much that we can go on whole genome sequence in any case. So in my opinion, the need to use gene panels or whole exome sequences will be out of date in a short time, because the cost is, the information loss, in my opinion, is not justified by the similar cost of the two technologies. What is the difference between short and long reads? Of course, as I mentioned you before, short reads have higher accuracy and higher throughput, whereas long reads have the cost of a higher error rates, but technology are improving and the error rates is continually decreasing, but have the enormous advantage of higher connectivity. So long reads have the advantage to give us the possibility to fully reconstruct a human genome that, as you know very well, is highly repetitive. And this is particularly true if you see in this slide, where you can see that in the human genome, there are about 300 mega base pair, which are refractory to sequencing. So they're very repetitive. And in this region, we have a very low coverage. And to identify even single point mutation in this region is very complicated. So in this sense, this means that we may lose some causative mutation if we are not able to reconstruct also this region. The problem, of course, can be solved by the use of long reads that, as I said before, are more error prone and even a little bit more expensive than short reads. So the problem of genome assembly is explained in this simple slide. The difference is to reconstruct a puzzle. The difference between a commercial puzzle is that we have an image on the cover that, of course, with the genome not have. And the other difficulty is that, by definition, a puzzle that we used to play as unique piece. This is, not, of course, not true for the human genome, where half of it is repetitive. So as you may understand very clearly that the longer there is, the better is the possibility to accurately reconstruct the full genome and thus to identify reliably all the variation. So now we go to what we can do, how genomics enter healthcare. Of course, as our blueprint is encoded in our genome, a full understanding of the information embedded in our genome is able to provide us information on many areas of disease. You can see a rare disease, complex disease, infection disease, and others. And so to enter what we call, of course, precision or personalized medicine. So this enterprise has been very clear to the European community, to the European Commission, and so EU promoted the so-called 1+ Million Genome Initiative. What is this initiative? Has been promoted by 24 EU member states, including also UK and Norway, who signed in 2018 the 1 Million Genome Initiative in order to go toward the construction for creating a European data infrastructure for genomic data, in order to implement shared rules that, of course, include GDPR, ethical, legal aspects, in order to have a real transition in the healthcare by the use of genomics. And this is also part of a bigger enterprise that is in the EU's agenda for the digital transformation of health and care that is under the umbrella of the so-called European Health Data Space Initiative. That what you can mostly understand that is the electronic health record that in many countries is now being established. So you can imagine what this means at European level if you want to study a rare disease, if you can share in a secure way the genetic data in order to tackle this kind of problems. So the final goal in this respect is to build the so-called Genome Data Infrastructure that is has been funded by the GDI project that aims to establish a federated, sustainable and secure technical infrastructure. So there has been a first project, the so-called B1MG project, that was the design study, and then the GDI project in order to establish the technical deployment of the IT infrastructure. And at the European level, Elixir, that is the European Research Infrastructure for Life Science Data, has been engaged by EU in order to lead this initiative. This is a project that will end next year, started in 2022, funded by 40 million euro and includes 22 countries, including Italy. Which are very briefly the component of GDI. The component of GDI you can see here. There are tools for data discovery, and state-of-the-art tool is the Beacon. There are tools to access in a secure way the information, like LSAI or REMS. There are tools for data processing that need to be reproducible, like the Docker or containerized system and federated compute. There are tools for data reception, for instance, the decryptation tool established by the Global Alliance for Genomic and Health, and of course, facilities for storage. And the chosen facility is the so-called Federated European Phenome Archive. So the storage of the data is no more centralized in a single country, but each single country hosts its own data, and this is, of course, needed by the national law and by the legal implication, but all these data are federated in order then all the metadata are accessible through a centralized system. And the Italian instance of GDI is now ready for use. So in practice, any user could eventually upload, do what is called data injection in the system. But in order to become, to enter production, we need formal endorsement and support by the Ministry of Health of Italy, with which there are now discussion ongoing. Another important project that I would like to mention is Genome of Europe. This other project is very needed, is, as you can see here, is funded again by the EU. Of course, we need the genetic background of the European population. This is very needed in order to assess if a genetic variant is rare or not in that specific population. And so this is another big enterprise that started recently and should end in 2028. The final goal will be to sequence 500,000 European citizens, but this project is address the first 100,000 genomes in order to provide the initial background of the European population. The Italian component of Genome of Europe is particularly relevant because I was not aware of this. Italy has a legal level of ancestry diversity that is due to the Roman Empire, because in Rome many population arrived. So as you can see in this PCA plot, we have a very, very high diversity in the Italian population. So the genetic background is really relevant in order to provide a correct assessment of the genetic variation. And I will show you an example shortly. This also because what we use now with a reference genome, many of you know the HG38 reference genome, is the results of a human genome project where there were some volunteers that were recruited in the New York State, as you can see at the end of 19th. And of course, this presents a relevant sampling bias in order to compare European and particularly the Italian population. And so at the moment, this is a very important problem, as you can see in this manuscript, in this paper published in the New England Journal of Medicine, because the sampling bias may lead to genetic misdiagnosis. And for instance, at the moment, over 70% of European samples are Finnish or British. So they really do not represent the actual European population with all its different countries. And this is the plan that we have for Italy. The plan is to generate about 15,000 whole genome sequence with 30-fold coverage. The two sequencing centers involved in this project will be Human Technopole in Milan and the omics facility of Elixir Italy, located here in Bari, across two institutions that are National Research Council and the University of Bari. In this plot, you can see the ethnicity of the samples for each country to contribute to Genome of Europe. Of course, this is particularly important because, as I mentioned before, even if two individuals are on average 99.9% identical, still they have many differences that are 4-5 million SNPs, from 500,000 to 1 million indels, about some 10,000 structural variants, a bunch of copy number variation and few de novo variants. Just to show you an example of the bias that can be introduced by the reference sampling bias, we carried out a few time ago during the pandemic exome sequencing of about 119 COVID-19 patients and some controls in order to identify the so-called loss of function rare variants. Using the whole collection of public data for the genomic background, we detected over 400 rare variants that decreased sharply to only 16 using as a control the Italian genetic background. So this just gives you an idea of what can mean to use the correct genetic background in order to discover causative rare variants. Now I will finish, give me other 5 minutes to give a few words on what is Elixir. Elixir is one of the research infrastructure promoted by the EU Commission through the so-called ESFRI, European Science Forum for Research Infrastructure. It has a hub-and-spoke structure with the hub in Wellcome Trust Genome Campus in Cambridge. At the moment has 23 national nodes, including, of course, Italy. You can see here the service provided by Elixir, database training tools, data standards, compute reports, and of course, scientific know-how. Elixir Italy is led by the National Research Council and at the moment has over 30 institutions that include university, other research centers, or technological provider like Sineca in Bologna or the National Institute of Nuclear Physics or the GAR network facility and so on. And the difference with respect to the European structure is the fact that in addition to the 5 platforms that I mentioned before, we also have the so-called omic platform, which has a rich portfolio of advanced platform for omics data production. This allows us to provide service to the research community, several genomic services. You can see here very briefly which are the services that we are able to provide with some of the advanced instrumentation we have. And of course, this is paralleled by a very powerful compute platform, which has its headquarters in a data center here in Bari, but is federated also with other compute and storage platforms across all Italy. One of the services we provide are virtual machines to analyze in a secure and legal compliant way the genomic data that can be provided to users that have this kind of necessity. And we also provide a data biorepository that can be used for storing the data, even sensitive or not sensitive. I don't need to show you which can be the application of GAI and Genome of Europe. Of course, we will have very powerful tools for analyzing non-communicable disease, of course, through polygenic risk scores or pharmacogenomics, or in the case of rare disease, to design tools for gene therapies. So this is the last slide. So we are just experiencing now an amazing revolution in precision medicine with the concurrent provision of omics data explosion and AI explosion. So I think we are in a very critical moment that, of course, can provide a real revolution and important impact on the health system for Europe and, of course, at international level. Of course, this is the result of very large group working on several aspects, and I think I can finish here. And if there are any questions, I will be happy to answer. Thank you very much.
Thank you, Professor Pesole. We can take, if there are immediate questions, immediately also, well, we can take them now. Otherwise, you can, so I just see, just raise your hand. So everyone is welcome here to ask questions. Please raise your hand if you have a question. If there are no immediate questions, Dr. Pesole, would you agree that we continue with the second presentation and everyone is keeping in mind and maybe writing down the questions, so they will be also...
I agree, and I can also share my presentation in case someone can, if you want, you can put this available for who is interested, okay?
That would be fantastic. Thanks a lot. I have already some questions, but maybe then we keep them, I will keep them for the end, and I would then hand over to Dr. Fosso for the second part of this webinar. Thanks already, Professor Pesole. It was very interesting, and thanks for the great overview.
Thank you.
Okay, can you hear me?
Perfect.
So, good afternoon. This will be the outline of this presentation. We start with some introduction about the concept and of microbiome and metagenomics, and then we will focus on the context application of ML and AI application on microbiome data. And then I will show you some application of ML and AI data on microbiome data by integrating data coming from different sources, but using the same layer. And then finally, multi-omics integration with a really interesting example. Before starting with the presentation, I would like that some of the pictures that I'm going to show are produced by using AI, but all the scientific content of the presentations was completely checked by me, directed by me. So I'm going to start with a provocative question. What does it mean to be human? So we know that in almost our cells in our body, there's the same content of nuclear DNA. We know that about the 2% of this genomic DNA is coding from protein, but actually the complexity of the cells that are that our genome is able to codify is related to the complexity of the transcriptome and the regulation of gene expression and post-transcriptional modification. So actually, in our body, we can find about 200 different cellular types, accounting for about 10 to 13 cells. So just to make it clear how large is this number, you have to think that actually in our galaxy, the Milky Way, it is estimated there are 10 to 11 stars. So actually in our body, in the body of each people in this room, there are let's say 100 cells more than stars in our galaxy. But if you think that is a really large complexity, you have also to think about that since our birth, our body is colonized by a large and a complex community of microbes. And when we talk about microbes, we refer to both prokaryotes, small eukaryotes, and also viruses. And an estimation about the number of cells that are able to colonize our body is around 10 to 14 cells. So it means that in our body lives 10 cells more than our genome is able to produce. So these cells are not just hosts on our body, but they are able to influence our physiology and are fundamental for our health. Actually, we know exactly that the effect of the Western lifestyle has modified during the last 15 years the composition of the gut microbiome, which is actually the most important microbial community living in our body. And the result of this change is a loss of biodiversity and loss of function, and the effect is the emergence of chronic disease such as allergies and so on. So it is also clear that it gives us a large repertoire of protein coding genes, estimated about 5 to 8 million protein coding genes that are, and these microbes are also able to work with our immune system, both the adaptive system and the native one, by modulating him and reducing the effect of inflammation. Of course, it is not stable during our life, so it changes according to different parameters such as ages, delivery mode, and actually we know that people born with cesarean delivery has a higher risk to encounter pathologies such as asthma or chronic disease. Diet or geographical origin, gender, the usage of drugs, in particular antibiotics, and also stress related to work life or daily life. So we need to take care about this microbiome because it is really crucial to regulate our health. There are several ways in which the microbiome is able to modulate and to improve our health status. And in particular, one of the most relevant action is the colonization resistance. So it is able to reduce the ability of some pathogens to colonize our body. And it is able to do that by using two different ways. The first one is a direct competition in which microbes that are normal components of our microbiome, and in particular normal components of the gut microbiome, are able to produce some antimicrobial peptides called bacteriocins that are able to avoid the colonization from some specific pathogenic species such as Citrobacterodentium and Salmonella enterica tifimurium. But at the same time, by eliciting the response of the native immune system, it simulates the production of antimicrobial molecular molecules, directly from cells of our bodies, in order to reduce the capability of colonization of some specific species such as Enterococcus faecalis. At the same time, it is also able to interact with our adaptive immune system, and it creates a tight collaboration between the immune system. Indeed, we know that the EGA secreted in the gut lumen are critical to control the proliferative capacity of these microbes in one way, but in another way, it allows them to colonize the mucosal substrate of our gut. Furthermore, these IgA are also able to control and counteract the activity of potentially pathogenic species. Another point that is crucial, and that is this is the crucial point that is missing in most of the pathologies related to the Western lifestyle, is that the lack of the biodiversity in our microbiome gut is reducing its capability to prevent an hyperactivation of our immune system. This is the key point that drives then to chronic inflammation. So the element that I would like to stress is that we are not just discussing about only one microbe, but we are discussing about a community of microbes, a really complex community of microbes composed by different types of cells, from prokaryotes to fungi, but also viruses. Just to stress the fact related to the community concept, actually we know that a microbiome that is completely balanced with the host is a microbiome in dysbiosis. But the lack of this state that leads to dysbiosis is the result of the perturbation of the overall ecology of the community. It is interesting to know that one of the most resistant infection, and in particular the infection by Clostridioides difficile, is usually treated by using fecal microbiome transplantation. It means that the result of the infection is an overall change in the microbial community living in our gut. So in order to eradicate Clostridioides difficile, it's required to use and to transplant a normal, let's say, a normal community that is able to counteract a specific action. On the other hand, there are some probiotic species, such as Bacteroides theta-iota-micron, that are able to do things that are crucial to generate a normal and stable community. Indeed, if from one side it promotes the formation of tight junction between the cells in our gut, but on the other hand, it promotes the immune system tolerance to normal components of the microbiome, and it digests fibers that we are not able to digest and produce short-chain fatty acids that are crucial for our health. So, just to stress again this concept, we are not just talking about a single microbes, but a community of microbes that are able to maintain our health during the lifespan. So actually, during the last century, there was a shift in the microbiology paradigm. So we moved to focalize our attention on single species or some single microbial species or on single pathogen species, and actually we know that these pathogens are just a tiny part of the whole microbial biodiversity, and we move to focus on the ecological concept related to microbial communities. And this ecological complex, this concept is strictly related to the abiosis and dysbiosis concept. So if this microbial community lives in balance with the host, so it's able to maintain to control the host immune system and counteract overstimulation of the immune system, and are able to produce molecules that are crucial for the health, such as short-chain fatty acids, this condition is called eubiosis. The lack of this condition, the colonization of pathogen species, of potentially pathogenic species, drives to inflammation, drives to diseases, and this is called dysbiosis. So, from an operational point of view, we can define the microbiome as the overall set of microbial species living into a specific biological niche or biome and its theater of activity. It means that not only the genes, but also the signals that are able to share among them and with the also, for instance, proteins, metabolites, and so on. When we focus on the microbiota, we are just focusing on the, let's say, bacteria, archaea, fungi, and protists that are living in our specific biome. And when we refer to the metagenomes, we are just referring to the overall set of genetic elements that are present in a specific sample. So, as I said before, during the last century, we observed a shift of the paradigm from a single species, from the genomics to the metagenome, so from a single microorganism to the community of microorganisms living into a specific environment. And this shift was mainly driven by two observations. The first one is made more about 14 years ago by Stalin Konopka, that coined the term the Great Plant Count Anomaly, in which they were able to understand that about 99% of microbes living in soils and water are not cultivable. So they are not investigable by using standard approaches. Then, in 1998, John Handelsman used for the first time the term metagenomics. She was interested in studying the antibiotic resistance potential of microbes living in soil. So they just extracted whole the DNA from soil samples and then used that DNA to prepare some expression vector to test the presence of some specific antibiotic resistant genes. So the idea was quite simple. quite simple. Instead of trying to cultivate these microbes, knowing that about 99% of them are not able to grow on a standard condition, we are just trying to verify if we are able to extract and identify antimicrobial genes. Of course, the advent of next generation sequencing technology in 2004 allows to reduce the sequencing cost and boost the application of metagenomics for the investigation, not only of the human microbiome, but also of the microbiomes living in different environments. Indeed, now the bottleneck is not the data production, but becomes the data analysis. So we need tools and we need appropriate computational framework in which we are able to analyze terabytes of data. From an experimental point of view, we can define two different metagenomics approach. The first one is the functional metagenomics that actually was the one proposed by Joe Endesman. And the second one is the sequence-based metagenomics. And in this case, we use sequencing. So we sequence the DNA or we sequence the RNA with the aim to understand which is the taxonomical composition of the microbial community. So we would like to know who are there, or also to try to infer what are the functional capabilities of these microbes. Actually, there are two approaches that are available: DNA metabarcoding and shotgun metagenomics. So in order to make it simple and quite straightforward to understand how this approach works, let's imagine our gut microbiome like a supermarket. And you would like to understand what are the legumes in this supermarket. So if you would just like to know what are the different legumes in our supermarket, you can simply scan the barcode on the different pockets. So actually, by using the barcodes, you are able to understand what are the products. So, and this is actually how meta-barcoding works. It leverages the availability of some universal marker genes, such as the 16S rDNA genes for prokaryotes, that can be easily amplified and sequenced, and then analyzed to infer the taxonomic composition of a specific sample. But on the other hand, you could be interested to understand not only what are the legumes, but all the products available in specific supermarkets, and you would like also to know what are the specific features of that products. So in that case, you cannot just use the barcode, but you have to extrapolate, read all the labels on the products. So in this case, we are talking about shotgun metagenomics, in which we extract the whole metagenome, so the whole genetic content presented in a specific environment, and then we randomly sequence all the genetic information. Of course, DNA metabarcoding is cheap in terms of sequencing and bioinformatic analysis, but it is less informative because we are just using short target sequence from 300 to 1.5 thousand base pair in case of long read sequencing, to understand what are the taxa present in a specific environment. And considering the higher plasticity in prokaryotic genomics, it is really hard to understand what are the species, and in most of the cases we are just able to understand what are the genera. So just to be clear, we are not able to say that in the specific sample there's Pseudomonas aeruginosa, but we can say that there's the Pseudomonas genera. Why? With shotgun metagenomics, we are also able to obtain a species, or in some cases, strain level classification. We are also able to reassemble full genome and characterize also the functional capabilities of these genomes. So, which is the context in which this presentation was made? So the idea is quite simple. Actually, we have a large amount of publicly available data sets that can be used to train AI and ML-based models. Furthermore, there is an increased interest in the multi-omics application. So we are able to obtain multi-layer characterization of the same environments. Another stuff that is quite interesting to understand is that usually this kind of data are high dimensional. So it means that we have a lot of features, and standard statistical approaches are not able to completely understand what are the difference between, let's say, healthy and non-healthy sample. So in this case, ML and AI became so useful to analyze this kind of data. As I said before, actually we are able to produce different layers of the same biome, so starting from metagenomics to metatranscriptomics, or also to understand what are the metabolites that are shared between these microbes with metabolomics. And we are also able to integrate metadata, so data, contextual data that are associated to specific sample, such as health condition, antibiotic usage, and age, gender, and BMI, and other metadata that can be useful to understand how microbial community changes according to the variation of specific data. Now I'm going to show you some application of machine learning on human health data. I will start with this example. This is an application of random forest, a specific tree-based approach that we use in 2022 to understand what are the main differences between healthy tissue and paired breast tissues in 33 women. We use standard statistical approach, and we found out that there was some specific species that were that changes between healthy and non-healthy tissues. Then we trained a simple random forest model, and this model was interesting because it was able to reach an accuracy about 89%, and it was really interesting to note that the most relevant feature that the model that contributes to the model accuracy was the Propionibacterium acnes, actually known as Cutibacterium acnes. This bacterium is a common component of the skin microbiome. It is really relevant because it has an action counteracting potential colonization by pathogens, and as is possible to see to observe in this heat map, its abundance was highly related with healthy samples. So the lack of Propionibacterium acnes could be used as a possible marker of tumoral tissues. This was a really recent paper by a mixed group from China and Japan, in which they use a different approach to try to understand what are the drivers of healthy and non-healthy microbiomes. Probably you know the paper by the work group working on describing the enterotypes. So enterotypes are just genera that are associated with different state of condition. With Prevotella associated with healthy states, and Ruminococcus and other genera associated with the less healthy states. In this kind of approach, these researchers use a different way to try to characterize the microbiome. They develop a complete framework called WireDancer, in which shotgun metagenomic data are analyzed in order to understand not what are the species, but what are the ecological guild that are associated with healthy and non-healthy samples. So again, we move our focus from a single species or a single group to a network of species. And in particular, they found that guild that were overrepresented with Bacteroides uniform are associated to disease, while yield that are overrepresented with the Prepotella copri are associated with healthy, and an intermediate states is present with network in which there is Bifidobacterium adolescentis. So again, we move from a single group of taxa or single genera to a complex idea of microbial ecological factors. Now I'm going to show you two papers in which we integrate data coming from different sources. In particular, we have, in this case, 16S data, so the DNA metabarcoding data, studying different disease associated with the autism spectrum disorders, coming from different groups in which we use the same bioinformatic analysis approach, and then we integrate data in an approach that we can see later called herd integration approach, to try to generate a larger data set and train machine learning models and use XAI, so explainable artificial intelligence applications such as SHEP in this case, to understand what are genera that are associated with ASD and healthy samples. We train different machine learning models, in particular random forest, a support vector classifier and a gradient booster classifier. And based on the best performing models, we were able to understand that Alloprevotella and Parasutturella were respectively associated to SD and healthy samples. So in this case, we integrate the same kind of data, but coming from different resources. One of the most difficult things to do in microbiome data analysis is try to understand what are the determinants of a healthy gut microbiome and a dense body gut microbiome. In this specific case, we focus not just on the prokaryotic microbiome, but we focus on the fungal microbiome, so the so-called microbiome. In particular, this is a work done by Giuseppe Fazzio, a researcher working in our group. We integrated data coming from more than 1,000 human gut samples, and we train a random forest model, and we also integrate this information with the result coming up with other different machine learning approaches relying on linear discriminant analysis and general linear models. And we found out that our random forest model was able to correctly classify healthy and healthy samples with an accuracy over 80%. And there was a really relevant overlap between the different methods in which 10 genera were found associated with, were relevant for models classification regardless the used approach. Now, last part of the presentation will focus more on the multi-omics data integration. As I said before, with microbiome data, there's a recent trend in which the researchers are trying to use information coming from different layers. So, for instance, from metagenomics, metabolomics, and host environment information to try to understand what are the drivers associated with specific disease or the healthy condition. The idea is that instead of using just one information, let's say taxonomical composition coming from DNA metabarcoding or shotgun metagenomics, integrate this information with other data, for instance, metaproteomics or metabolomics. Of course, the framework related to multi-omics data integration is quite complex because different types of data has different ways to be treated. For instance, as I said before, metagenomics data are compositional, are really sparse, are zero inflated. So we need to treat them really careful, and we need to use specific approach to take into account the compositionality of this data. The same careful procedures should be used also for the other types of data, especially if you are working with the metabolomic and metaproteomic data. But the key idea of these approaches is that by leveraging information coming from different layers, we are trying to understand if you would like to use a supervised approach. So we will use a classification approach to understand, let's say, what are the difference between healthy or non-healthy samples, or we would like to infer, for instance, what are the taxa or what are the metabolites that are associated with glucose level in blood. In this specific case, we are a regression task. Again, we have to face with the peculiarities of this kind of data that are compositionality in case of metagenomic data, batch effects, and it is really relevant when you have to try to integrate data coming from different resources, even if they analyze the same layer. For instance, if you are trying to analyze metabarcoding data, there's an effect related to different laboratory practice that results in noise in your data. So you have to take into account this batch effect to avoid that the classification is driven by not feature related to a specific condition, but features that are affected by technical issues. And of course, as I said before, sparsity and zero inflated table are related mostly to the methodological approaches, in particular when we use PCR for DNA metabarcoding data generation. We have tables that are inflated by zero just because there are species that are not present in our samples or species that were not amplified due to PCR failure. Of course, when you move to into the multi-omics data integration, there's another issues related to data missing from specific layer. For instance, you have 100 sample produced with DNA metabarcoding, but for just one sample, you don't have the second layer related to the metabolomics data. In that specific case, you have to decide if that sample is relevant or is it better to discard the specific sample because you don't have enough, information about the metabolomic layer. So how we can perform this kind of integration? Speaker in 2008 divided the integration approach in three principal ways: early integration, intermediate integration, and late integration. Depending on the data that they are going to analyze, this kind of approach can be more or less reliable. In early integration, we are just going to concatenate features from the different layer and then perform a unique integration analysis to obtain the output. In the intermediate integration, you are not working on your features, but you are working on the latent features, or the features that are results from a specific transformation of your original features, and then the integration model is trained on just on this feature. The last approach, which is really interesting and is quite straightforward to understand, is that different layers, such as DNA metabarcoding, metabolomics, metaproteomics, are analyzed separately. Then features that are relevant for, let's say, ML model or classical statistical approaches are finally integrated into integration model. Just to make an example that is quite relevant, that is quite new in the microbiome data analysis, there's a tool called MindT that is specifically designed for that integration of microbiome data. It relies on a sparse generalized canonical correlation analysis. Just to make it clear, imagine that it tries to find correlation between the different layers for instance, DNA metabarcoding data and metabolomics data, and try to maximize the correlation between these modules, so these highly correlated features from different layers, from the reference classes, such as healthy and non-healthy. MIND-T was evaluated on 9 different metagenomic datasets, and the robustness of these methods was demonstrated by the highly performance in discriminative power between healthy and non-healthy on 9 different diseases obtained by the modus extracted by the tool. So it is really able to extract features, let's say a group of features, model features that are really able to discriminate between 2 different conditions. So in conclusion, data integration is crucial not only to improve the prediction, let's say to discriminate between 2 conditions, but it is also relevant to create, to understand what are the biological community, the biological fields that are associated with specific condition or driving to specific pathologies. So we can improve our understanding of the microbial world and how this world is relevant for our life and our health. So thank you for your attention and open for questions.